Hello everyone, today I'll be talking about the rolling circle replication and the inhibitors of replication. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. The DNA replication occurs in all living organisms, acting as the most essential part for the biological inheritance, as it ensures that each of the new cells receive its own copy of the DNA. It is semi-conservative in nature. The DNA molecules which can replicate autonomously are known as replicons. Example, plasmids, phage DNA, chromosomes. Replicons have at least one origin of the replication. The rolling circle replication is one of the most well-characterized model of DNA replication. It was first described by Gilbert and Dressler and Asen Pereira de Silva and Jacob in 1968. It is called the rolling circle replication because it was first discovered in a type of phage where the template circle seems to roll. It was described while analyzing the replication of E. coli bacteriophage 5X174. Since then, many other genetic elements replicating by the rolling circle mechanism have been identified, including bacteriophages, plasmids of gram-positive and gram-negative bacteria, archaeal plasmids, and eukaryotic viruses. The enzymes involved in the rolling circle replication. First one is the Rep-A protein. The Rep-A protein exhibits the nucleolytic activity and they cleave the leading DNA strand to initiate the replication process. The active sites of the enzyme contain conserved tyrosine residues that are directly involved in the catalytic function. It is encoded by the plasmid gene. Then we have the DNA polymerase enzyme, which is involved in the elongation process. It results in the formation of a new strand. Helicase enzyme unwinds the DNA strand, whereas the ligase enzyme is responsible for the sealing of the nicks. Then we have the mechanism of the rolling circle replication. Rep initiation protein interacts with the two DNA regions called the bind site and the nick site located within the double-stranded origin. The initiator protein interacts with the bind locus in a non-covalent manner. The exposed nick site of the double-stranded origin is cleaved by the catalytic tyrosine of initiator protein. Now we have the 3' end with the presence of hydroxyl group and the 5' end with the presence of phosphate group. Rep protein will remain attached to the phosphate at the 5' end of the DNA. The DNA polymerase III, which is the replicative chromosome, uses the free 3-hydroxyl end at the break as a primer to replicate around the circle. Rep initiation protein recruits a helicase to unwind. As DNA unwinds, it is coated by the single-stranded binding proteins. As replication proceeds, the nicked strand continues to be coated by the single-stranded binding protein. The single-stranded binding coated strand peels off until the replication of the intact strand is complete. The two ends of the nicked single strand is rejoined by the Rep-A protein and released. DNA ligase seals the nick in DNA double-stranded molecule. The single-stranded DNA can now be replicated. A region of the DNA becomes looped, allowing the RNA polymerase access to the DNA to form a primer. The host DNA polymerase uses primer as a starting point for the synthesis of the DNA. Finally, the DNA ligase seals the remaining nick, resulting in the plasmid, which is double-stranded. In this, we can see that how the Rep-A protein cleaves the double-stranded molecule. It results in the formation of a nick, and how the DNA polymerase III recruits or results in a formation of a new strand. The DNA ligase seals the nick, resulting in the formation of a double-stranded molecule, a single-stranded molecule, and the release of the Rep-A protein. Then our rolling circle replication mechanism of 5X174 and PT181 differ significantly at leading strand termination stage. This is the consequence of the different life strategies of bacteriophages and bacterial plasmids. The best known rolling circle replication replicons are bacteriophages and the bacterial plasmids. In Psi-X174, the termination of the leading strand replication initiates another round of replication, enabling the phage to rapidly amplify its genome before lysis and escape from the bacterial cell, whereas the PT181 copies per bacterial cell is tightly regulated. Therefore, after a single replication cycle, Rep-C, the replication initiator protein, is inactivated and cannot initiate another round of the replication. The significance of the rolling circle replication. Interestingly, rolling circle replication plays a vital role in the horizontal gene transfer. Horizontal gene transfer contributes greatly to the plasticity and evolution of the prokaryotic and eukaryotic genomes. It is also thought that some transposable elements such as the bacterial IS91 like insertion sequences and eukaryotic helitrons use PCR to replicate in the host genomes. According to their biological roles, the rolling circle replication initiator proteins are divided into three groups: the Rep-A proteins, also known as the vegetative replication proteins, the MOP proteins, which are responsible for the mobilization in the conjugal transfer, and the TNP proteins, that is, the transposition proteins. Now we will be talking about the inhibitors of replication. There are several substances which inhibit the RNA primer synthesis as well as the DNA elongation and thus interfere with the DNA replication. Inhibition of replication also prevents the cell division. The inhibitors of the eukaryotic replication can be used as the anti-cancer drugs, and there are many substances which can be used as the inhibitors of the replication. The sum of the inhibitors which we will be talking about are actinomycin D. Actinomycin D intercalates into the double helical DNA between successive GC base pair, thus deforming the DNA. The elongation of the RNA chains by RNA polymerase in both bacteria and eukaryotes is specifically inhibited by the antibiotic actinomycin D. This local alteration prevents the movement of the polymerase along the template. In effect, the actinomycin D jams the zipper because actinomycin D inhibits RNA elongation in intact as well as in cellular extracts. It has become very useful for identifying the cell processes that depend upon the RNA synthesis. Then we have the acridine. Acridine inhibits the RNA synthesis in the similar fashion like actinomycin D. Quinolones are a key group of antibiotics that interfere with the DNA synthesis by inhibiting the topoisomerase, most frequently topoisomerase II, an enzyme involved in the DNA replication. Cisplatin is the chemotherapeutic drug. It is an anti-cancer drug which produces 1,2 intrastrand crosslinks of purine bases with cisplatin, thereby preventing replication. Ciprofloxacin interferes with the DNA breakage and rejoining process. Rifampicin binds specifically to the beta subunit of the bacterial RNA polymerase. Thus, DNA replication is inhibited by affecting the RNA primer synthesis. Then we have the novobiocin and nalidixic acid, which inhibits the elongation by inhibiting DNA gyrase, which functions as a replication swivel. Afidocolin inhibits activity of the DNA polymerase 1, 2, and 3, as well as the alpha, delta, and epsilon of prokaryotes and eukaryotes respectively. N-ethylmelamide inhibits the activity of the DNA polymerase 1 and 2 and eukaryotic DNA polymerase gamma. These are my references. Thank you.